Hello, good morning, and you're welcome back. Today we are going to continue our discussion. Now we are discussing techniques, microbial techniques, that we use in the laboratory in order to carry out our different experiments and investigations. We have a video where we have spoken about bright field microscopy. So please, go and look at it, like, subscribe, and try to follow us. Look at what we are doing. Today we are going to talk about phase contrast microscopy. That's the second technique we are talking about. So what's phase contrast microscopy? Now, phase contrast microscopy is a technique that we use to study cells, living cells. Is a microscopy technique. And it is just like, we can call it an improvement or a modification on the bright field microscopy. Because what we do in phase contrast microscopy is that we view specimens, living specimens, and we view them without staining those specimens. This is very important. When we talked about bright field microscopy, we said we apply different types of stains, your Giemsa stain, your Wright stain, all these different types of stains to our specimen. Now, an issue with the application of stains and dyes is that sometimes or most times it leads to the death of the specimen. So the living cell that you're studying, once you stain that cell, the stain or the dye kills that cell and then you view the cell. Now, there are situations when we want to view a cell even as it is alive. We don't want the cell dead. And in such situations, we cannot apply dyes or stains to such a cell because that will kill the cell. And we want to view the cell while it's alive. We want to look at some of the activities that the cell is carrying out even while it's living. So in order to do that, we do what we call phase contrast microscopy. And in this type of microscopy, what we do is simply to modify the normal light microscope. To modify the normal light microscope by adding, there's what we call an annular diaphragm. Now, this annular diaphragm, we add it to the condenser of the light microscope. We know that light, in the light microscope, you are using either your LED or your halogen lamps as a source of light, and then that light is being channeled through the condenser and it comes out to hit the image which is mounted on the stage. Now, in a situation where we now add an annular diaphragm to the condenser, and the idea is that we want to look at channeling that light source in such a way that it doesn't hit the specimen full glare. We want it to hit the specimen in such a way that we can have an image that is different. Now, it's important to note that many things in physics, there's what we call refractive index. So, if you are looking at a source of light, if it passes from one face to another face, you see that that source of light is sort of bends in its movement, in the way it is moving. And this is what we're exploiting when we do phase contrast microscopy. We know that different components of the cell have different refractive indexes. And because they have different refractive indexes, when you have a light source hitting that cell, all the different components within the cell, they refract the light at different angles, refract the light in different ways. And it's this difference in refraction that helps to create an image that we are able to use to study. Now, there's also something called a faceplate, and this faceplate is attached to our objective lens, any of the objective lenses that we want to use, the faceplate. So it's attached to the objective lens, and you have an annular diaphragm that is put inside the condenser. When you do these two things, you are changing a simple light microscope into a phase contrast microscope, where you now begin to study different cells. And the idea is that you're able to study cells while they are still living. And this is very important. It's important to note that a simple microscope, such as our Olympus microscope, our Nikon microscope, and all these microscopes that we use in teaching and research laboratories, are suitable. They're good enough to be used for phase contrast microscopy. You just need to add the faceplate and the annular diaphragm in these two different places that I've mentioned. And once they are added, you can now view these cells. You can view these cells in their living state. So, for an example, a fertility lab or a lab that is doing research on fertility wants to look at sperm cells, wants to look at how motile sperm cells are in order to see their viability and their ability to be able to, cause pregnancy or to, fertilize an egg. So you want to view such cells, and to view such cells, You don't want to stain such cells. You don't want to kill such cells. You don't want to apply dye to such cells. You want to view them in their living state. And to be able to view them in their living state, what you do is phase contrast microscopy, so that you can view them, you can see the tail of the sperm cell, you can see how it is moving, you can see the swimming action, and this is important in that kind of study. So these are important things that we need to know. When we look at some of the things that we can study in phase contrast microscopy, we can study bacterial cells. So if you want to study the way bacterial cells divide, binary fission, you want to study conjugation, you want to study all these different life processes that bacterial cells are involved in, and you want to see it real time. You want to see how it is occurring. You can use the phase contrast microscopy to be able to view all of this, and you can see it real time. For an example, if you want to see protozoa, you take pond water samples, take them to the laboratory, and then put them on your slide, mount them, and you want to look at different kinds of protozoa. You want to look at paramecium, you want to look at amoeba, or you even want to have blood samples and you want to look at your no, not blood samples now, water samples, because it needs clear samples. So you want to look at your paramecium, your volvulus, your chlamydomonas, all these kind of things. You want to view them even while they are in those pond water, and then you can use phase contrast microscopy, which will help you. You can use it to look at yeast cells. Yeast cells, as those yeast cells are going through budding, which is their own method of, which is one of their methods of reproduction. You want to view it and see it happening live. So to see that, you have to apply the use of the phase contrast microscopy. These are examples, and I've also told you the example of I've given you an example before of what you can use it to view. Also, if you are looking at cancer cells and you want to look at their structure, you want to look at how they divide, all these things, you can use the phase contrast microscopy. Now, what are some of the advantages of making use of the phase contrast microscopy? You realize that there's no staining that is needed. You do not need to stain the specimen. It is cheap, it is easy. Once you do have the equipment that you want to use to view, you can view such an equipment. So these are some of the advantages of using the phase contrast microscopy. But it also has some disadvantages. It's prone to a lot of artifacts. If you are used to using a microscope, you know that there are what we call artifacts. They look like dirt on the surface of the microscope. And if you are not trained, you don't have skills in using microscope, you may assume that there are some microorganisms or there are some important things. Meanwhile, they are just artifacts. So they are prone to artifacts. Then you can't use them for any thick cells or thick materials. Materials of living things that are thick, it will be difficult for the light to flow through them and for you to get an image that is responsible or that you can use. So these are some of the disadvantages. And we must understand the phase contrast microscope is important for you to help you to see cells as they are living and make use of them. Thank you for listening. Thank you for following us thus far. Remember to like, remember to share our content, and do have a beautiful day.